Glycolysis. It's the way that we take a high energy molecule such as glucose and break it down. Today we're going to learn about the preparatory phase of glycolysis as well as the payoff phase. So glycolysis starts off with the preparatory phase, and this is where we start to invest ATP in order to accomplish a specific reaction that we can get off later in the payoff phase of glycolysis. So, no surprise, we're going to start with glucose. So glucose can actually be shuttled out of the cell with relative ease. So one thing we have to do once we have glucose in the cell and we're ready to perform glycolysis is put a phosphate group on it. So that's the first step of glycolysis. We're going to go ahead and take hexokinase with an ATP molecule and use that with glucose in order to get glucose 6-phosphate as well as 1 ADP out. So we went ahead and moved that phosphate group from the ATP and put it onto the glucose. Now glucose is trapped inside the cell, can no longer leave, which is actually exactly what we want. While many might not say this is the commitment step of glycolysis, this is definitely one of our first steps. We're already proceeding down this path. In fact, hexokinase cannot be reversed. We actually need another enzyme to reverse that. So now that we have glucose 6-phosphate, we've trapped it in the cell, we can actually go ahead and run it with phosphohexosisomerase, which in this case, all we're going to do is go ahead and change that glucose 6-phosphate to fructose 6-phosphate. Now with glucose 6-phosphate trapped in the cell, we can actually go ahead and use the next enzyme, this one being phosphohexosisomerase. With phosphohexosisomerase, we're going to take our hexose sugar, glucose in this case, with the phosphate, and we're going to actually change that to fructose 6-phosphate. So at this point, we're going to go ahead and start step 3 of glycolysis. This is the second step so far that we've had to invest ATP, and we're going to go ahead and use phosphofructose kinase 1. And that's going to require an ATP molecule, as I've already stated. So we're going to use a ATP molecule and move one of those phosphate groups to form fructose 6-phosphate into fructose 1,6-bisphosphate. Phosphofructose kinase 1 is an irreversible step. An easy way to know this is it required ATP. We needed a large amount of energy to overcome our delta G, or free energy. This is highly unfavorable unless we had ATP. So we're going to need another enzyme like we talked about in gluconeogenesis to overcome this step when we're building glucose back up from our pyruvate. And that now moves us on to our fourth step of glycolysis, using the enzyme called atolase. Atolase is going to go ahead and do exactly what it sounds like. It's going to lyse our fructose 1,6-bisphosphate into 2 molecules, which is rather important for a plethora of other pathways in the body, but particularly for glycolysis, one of our products is now going to need to be converted into the right product. And so we're going to have our 2 products. First one being glyceraldehyde 3-phosphate, which is our main molecule that we're actually going to push forward for the rest of glycolysis. We're also going to get dihydroxyacetone phosphate. And this is important for several other mechanisms in the body. However, having 2 products from our 4th reaction of glycolysis actually will help our 6th step of glycolysis. Oddly enough, usually what's called the 5th step of glycolysis is an enzyme called triose phosphate isomerase, which ultimately is just taking DAP, or dihydroxyacetone phosphate, as we talked about earlier, and glyceraldehyde 3-phosphate, and all it does is change in between the 2. And ultimately, being able to control the concentrations of what we need, glyceraldehyde 3-phosphate, to continue glycolysis helps us with the Le Chatelier's principle. A quick review of Le Chatelier's principle in this sense is that as we control the concentrations of equilibrium, if we lower or raise one of those concentrations, we can actually force a reaction a certain way. So, coming back to glycolysis now, if we can control the concentration of glyceraldehyde 3-phosphate, we can ultimately control the 6th step of glycolysis. Now, it's really important to remember, because we had 2 products from atolase acting on fructose 1,6-bisphosphate with DAP as well as glyceraldehyde 3-phosphate, we now, for the remainder of glycolysis, have 2 of each molecule, or 2 of each substrate, or 2 of each product. This is important because not only are we just getting 1 ATP out for each step, we're going to be doing that twice. So actually you're going to get even more out from 1 glucose molecule. A key note to remember is from 1 glucose, you will get 2 pyruvate out. And so from this point on, we are actually going to be calling the payoff phase. We'll actually have 2 molecules for each one. Now, it's not necessary to go through each enzyme with 2 molecules, however, it is important when factoring out how much energy we're getting out of glycolysis. So, at the start of the 6th step of glycolysis, we're going to have 2 glyceraldehyde 3-phosphates using the enzyme glyceraldehyde 3-phosphate dehydrogenase. So in order to turn those 2 glyceraldehyde 3-phosphates, we're going to go ahead and need 2 inorganic phosphates, 2 NAD+, in order to get the product that we want of 1,3-bisphosphoglycerate. Alongside that, we're also going to get out 2 NADH with 2 hydrogen ions. This is important because NADH, it does have an ATP equivalence of 2.5 for each NADH. So in that step alone, ultimately getting out 5 ATP from our 2 glyceraldehyde 3-phosphates. And proceeding down glycolysis, we now are going to use the enzyme phosphoglycerate kinase for the 7th step, as well as 2 ADP. We're going to phosphorylate the 2 ADP into 2 ATP. So we're going to be pulling off 1 phosphate group from the 1,3-bisphosphoglycerate. Now the product of the 7th step is going to be 3-phosphoglycerate, which will move us into the 8th step of glycolysis, requiring the enzyme phosphoglycerate mutase, giving us the product of 2-phosphoglycerate. Phosphoglycerate mutase is really just moving that phosphate found on the 3rd carbon now to the 2nd carbon. It's a simple step, but ultimately will impact the free energy of the 9th step of glycolysis. In the 9th step of glycolysis, we're going to use the enzyme enolase. And in this step, we're actually going to get out 2 waters. And from this, our product will now be phosphoenol pyruvate. Phosphoenol pyruvate, also commonly called PEP, or PEP. And now we are on our last step of glycolysis. This is an irreversible step. In order to overcome this step now for gluconeogenesis, for pyruvate to now move back up to glycolysis, we're going to need new enzymes. However, for glycolysis, bringing PEP to pyruvate is going to require the enzyme pyruvate kinase. And in this case, we're going to take 2 ADP molecules and phosphorylate them to turn them into 2 ATP molecules, pulling off a phosphate group from each of the PEP molecules. And now, we have finally come to the end of glycolysis and hit pyruvate. This is exactly where we want to be, as we can now head into the citric acid cycle, and it's important to remember that our last step of glycolysis was irreversible. This is important. We don't want to lose our pyruvate. We don't want a reversible step moving our pyruvate back into PEP. In fact, we want to keep it pyruvate so we can now move on forward through obtaining even more energy through the citric acid cycle. So, a recap of glycolysis. Step 1, 3, and 10 are irreversible steps. These enzymes are hexokinase, phosphofructose kinase 1, and pyruvate kinase. What is often referred to as the committal step of glycolysis is step 3, which requires phosphofructose kinase 1. At this point, you are committing to go down glycolysis. While there may be other mechanisms that may use DAP, or dihydroxyacetone phosphate, ultimately glyceraldehyde 3-phosphate, one of the products of aldolase, our 4th step, will continue down glycolysis, and even some of that DAP will actually end up being turned back into glyceraldehyde 3-phosphate. So the majority of our product will continue down glycolysis. It is also very important to realize how much energy we actually got out of this. In the preparatory phase of glycolysis, we needed 2 ATP. In the payoff phase of glycolysis, we got 4 ATP along 2 NADH. So, as I mentioned several times while we were going through glycolysis, one of the ways that we can now take pyruvate back into glycolysis is called gluconeogenesis.